The overall goal of this procedure is to observe the fate of the replisome and RNA polymerase, or RNAP, following a head-on collision. This is accomplished by first assembling the transcription complex on biatenylated DNA and immobilizing it on streptavidin beads, which will result in the halted RNAP elongation complex. The second step of the procedure is to ligate pre-annealed forked DNA to the RNAP elongation complex to form a replication fork downstream of the head-on RNAP. The third step of the procedure is to assemble the replisome and initiate leading strand synthesis at the replication fork. The final step of the procedure is to isolate the DNA from the magnetic beads and purify the DNA over a PCR cleanup column. Ultimately, results can be obtained that show the replisome stalls upon collision with the head-on transcription complex through an alkaline agarose gel of the purified radiolabeled DNA products.
Hi, I'm Richard Pomerantz from the laboratory of Mike O'Donnell at the Rockefeller University. Today I will show you a procedure how to perform a collision of the replisome with a head-on RNA polymerase in solid phase. I use this procedure in our laboratory to study how the process of replication is affected by transcription complexes along DNA. So let's get started.
To begin this protocol, mix RNAP holoenzyme with a biatenylated 3.6 kb DNA template containing the T7A1 promoter in 100 microliters of buffer A. Incubate the mixture for 10 minutes at 37 degrees Celsius. After the 10-minute incubation, add ribonucleotides, adenosine triphosphate, cytidine triphosphate, and adenylyl uridine, which will limit RNA synthesis to 20 nucleotides. Incubate the solution for an additional 10 minutes at 37 degrees Celsius. To immobilize the halted RNAP elongation complex onto beads, add the solution to streptavidin-coated magnetic beads. Let the mixture incubate for 10 minutes at room temperature. Purify the immobilized halted RNAP elongation complex by washing the beads with 0.9 milliliters of high-salt buffer to remove nonspecific RNAP DNA complexes. Following each wash, remove the supernatant by magnetic separation. This wash should be repeated 5 times. Then the beads should be washed twice more with 0.9 milliliters of buffer A. After the final wash, the downstream replication fork can be assembled. To form a replication fork downstream of the head-on RNAP, resuspend the magnetic streptavidin beads attached to the halted RNAP elongation complex in 100 microliters of New England Biolab's buffer 4. Next, add 10 units of shrimp alkaline phosphatase, or SAP1, and incubate the sample for 10 minutes at 37 degrees Celsius. Wash the beads 3 times with 0.9 milliliters of buffer A. Then, resuspend the beads in 50 microliters of quick T4 ligation reaction buffer. Add 2 microliters of quick T4 ligase and pre-annealed forked DNA. Incubate the solution for 10 minutes at room temperature. Finally, wash the beads 3 more times with 0.9 milliliters of buffer A. Mix hexameric DNA B helicase with the magnetic streptavidin beads, which are attached to the halted RNAP elongation complex and the downstream replication fork. Prepare the solution in 150 microliters of buffer A and incubate it for 30 seconds at 23 degrees Celsius. Following the brief 30-second incubation at 23 degrees Celsius, add polymerase 3, beta-clamp, ATP, DGTP, DATP, alpha P32 labeled DGTP, and alpha P32 labeled DATP to a volume of 200 microliters. Incubate the sample for 5 minutes at 37 degrees Celsius. Initiate replication by adding single-stranded DNA binding protein, dCTP, and dTTP. Also add alpha P32 labeled dTTP and dCTP to a final volume of 250 microliters. After 10 minutes, terminate the reactions by adding 12 microliters of 0.5 molar EDTA. Next, boil the streptavidin beads to release the DNA from the beads. Remove any residual DNA by treating the beads with proteinase K for 30 minutes at 50 degrees Celsius. Boil the beads again and remove the supernatant by magnetic separation. Purify the combined supernatant containing the DNA using the QIAGEN PCR cleanup kit. Finally, analyze the purified radiolabeled DNA products in an alkaline agarose gel. Collision of the replosome with a head-on RNA-P usually results in two products of 2.5 kb and 3.6 kb in length. The 2.5 kb product represents the length of the DNA from the fork to the halted RNA-P. This results from replosome stalling upon collision with the RNA-P. The 3.6 kb product represents full-length DNA and results from either incomplete occupancy of the promoter by RNA-P or partial replosome read-through of the head-on RNA-P. A good result demonstrates that approximately 50% full-length DNA is produced. However, in some cases, less occupancy of the promoter by RNA-P occurs and a higher percentage of full-length DNA is observed. This is because a greater population of replisomes does not encounter a head-on RNA-P. Replication in the absence of a halted RNA-P results in only full-length DNA.
We've just showed you how to perform replication in solid phase in the presence of a halted RNA polymerase elongation complex bound to the DNA. When doing this procedure, it's important to wash the halted transcription complex with high salt to remove excess RNA polymerase, which nonspecifically binds to the DNA and inhibits replication. So that's it. Thanks for watching, and good luck with your experiments.